Assalamualaikum and hello madam. I'm Maryam, and in this video, me and my friends would like to present on the topic of molecular biology technique, regulation of the TRP operon. Firstly, the molecular biology technique demonstrated in this presentation is operon-based gene regulation. It is defined as a molecular biology mechanism used by prokaryotes to control the transcription of multiple functionally related genes under a single promoter in response to environmental signals. The TRP operon is a repressible operon that regulates genes involved in tryptophan biosynthesis, which is an anabolic pathway. Next, the TRP operon is found in E. coli and it controls the synthesis of the amino acid tryptophan. Unlike the lac operon, which is inducible, the TRP operon is normally on and is turned off when tryptophan is abundant. This is an example of a negative regulation, and this regulation helps the cell conserve energy by preventing unnecessary enzyme production. Additionally, in E. coli, the TRP operon regulates genes required for tryptophan biosynthesis based on intracellular tryptophan availability. For the next part, Farah will explain the structure of the TRP operon of E. coli and how tryptophan controls transcription through repression and de-repression.
Thank you, Maryam, for explaining the overview of TRP operon. Next, let's look into the TRP operon of E. coli that is organized into three main sections. First, the regulatory region that contains the promoter, where RNA polymerase bind, overlapping operator site that bind the tryptophan-activated repressor to inhibit transcription, and the leader regions, which include the attenuator and encodes 40 amino acid peptide with 2 tryptophan residue that determine whether transcription stop early or continue. Next, the structural regions that are arranged TRPE, TRPT, TRPC, TRPB, TRPA that encode 7 enzyme domain with 2 gene fusion present, which are TRPG fused to TRPD and TRPF fused to TRPC. There is also an internal promoter near the end of TRPD allows independent transcription of downstream genes. Lastly, transcription termination occur either at attenuator in TRPL or at the T tandem row independent terminator after TRPA, ensuring tryptophan is synthesized when only needed. Next, I will be explaining how TRP operon operates as a repressible system using repression and de-repression to control tryptophan levels. When tryptophan is abundant, it acts as co-repressor by binding to the inactive apoprepressor protein and activating it. The active repressor then binds to the operator, blocking RNA polymerase and preventing transcription, which stops enzyme production through negative feedback. When tryptophan levels are low, the repressor remains inactive and cannot bind to the operator, leaving the promoter accessible. RNA polymerase then can transcribe the fast structural genes, allowing the cell to produce the enzyme needed to synthesize tryptophan. Repression alone regulates expression over 80-fold range in response to the tryptophan reconcentration. Next, DINI will explain attenuation, which provides additional layer of.
Now let's move on to the second level of regulation called transcription attenuation. This is fine-tuning mechanism that allows the cell to control amount of transcription that occur even after RNA polymerase has already started. Attenuations happen in the leader regions of the operon and is depend on the availability of charged tRNA trip. This mechanism is possible. Why? Because in prokaryote, transcription and translations are coupled, meaning that translations of mRNA begin even while the RNA is still being transcribed. This allows the ribosome movement to directly influence the process of transcription. The leader regions of three operon contain 4 regions that can form different RNA structure, which is hairpin loop during transcription. This secondary structure play a key role in determining whether transcription will continue or terminated. The 2-3 hairpin form when tryptophan levels are low, allowing transcription to continue. The 3-4 hairpin loops form when tryptophan levels are high, causing transcription terminations early. The ability of this region to form hairpin loop ensures that the operon is transcribed only when needed. Here how attenuation works in detail. First, high tryptophan level. When tryptophan is abundant, charged tRNA chip is also abundant. The ribosome will move quickly around the mRNA, allowing the 3-4 terminator hairpin to form. This causes transcription to stop earlier, preventing the production of enzymes involved in tryptophan biosynthesis. Second, Low tryptophan level. When tryptophan is scarce, there isn't enough charge in tRNA trip, and the ribosome will stall at the tryptophan caudal in the ladder region. This allows the 2,3 anti-terminator hairpin to form, which will prevent the 3,4 terminator from forming, allowing transcription to continue and the production of tryptophan biosynthesis enzyme. This mechanism gives a cell a way precisely regulate transcription based on the tryptophan concentration. Now, I will pass it to the next member.
My name is BATRISYIA and I will be discussing the relation of the Tryptophan regulatory mechanisms to the research paper as well as its biological significance and conclusion. According to the study by Janowski in 2013, the Tryptophan in E. coli is regulated by two main molecular biology techniques. The first is repressor-mediated transcriptional repression, where tryptophan acts as a co-repressor to block transcription initiation. The second mechanism is attenuation-based transcription termination, which responds to the level of charged tRNA for tryptophan during transcription. Together, these two regulatory mechanisms control Tryptophan expression over an approximately 600-fold range, ensuring efficient regulation of tryptophan biosynthesis. This tight regulation prevents unnecessary enzyme production when tryptophan is abundant, which helps conserve cellular energy. It also ensures amino acid homeostasis by allowing rapid synthesis of tryptophan when intracellular levels are low. Because of this efficient control, the Tryptophan is considered a classic model for studying gene regulation in prokaryotes. In conclusion, the Tryptophan is a repressible operon that is directly controlled by tryptophan availability. It uses negative regulation and attenuation to precisely control gene expression in bacterial cells. And that concludes our presentation on the regulation of the Tryptophan in E. coli. Thank you for watching.